with us today to explore the frontiers of science and medicine. So, to officially commence our program, it is our great honor to invite the President of Asia University, Dr. Jeffrey Tai, to deliver the opening remarks. President Tai has been a visionary leader in promoting academic excellence and fostering international collaboration. So please join me in welcome President Tai to the stage. 让我们热烈欢迎校长致辞。
Professor Südhof, Mr. Morowitz, and also our brother university, former president of China Management University, Huang, and then the superintendent, Liao, the China Medical University Hospital, and also the Professor Chen too from the Pharmacy College of China Medical University, and also we have the academic Sinica, Director Cheng, and also the Director Huang from National Taiwan University. And ladies and gentlemen, good morning.
Good morning.
Yeah, after yesterday, we have an excellent talk by Professor Südhof, excellent speech, and also a great dialogue with the audience. So I consider a very successful event yesterday. Thank you again. Give a big applause to Professor Südhof. Truly master the care of the science, education, and the humankind of future. And today we're going to discuss a very important issue. As we know, over 58 million population suffer neurodegenerative disease, and so we need how to take care of those people. So we have to develop drug, but develop drug is very expensive. Normally, developing drug it costs $1 billion. One maybe 2-3 billion to 6 billion, very expensive, and take 10 to 15 years, and only 10% death successful rate. So it's very challenging, not a scientific problem, and also how to do patient care, a lot of problem. So I'm very grateful that Professor Südhof bring up this topic. Actually, that's the talk, original plan to give a talk yesterday, but he decided to give a more general talk yesterday. Also, very important talk about the integrity in our culture, in our community culture. It's very important topic, and thank you all the excellent point advice to us. And today we have a very distinguished panel, including Professor Südhof, and also the superintendent, this Liao, Liao, the China Medical University Hospital. He also doing a lot of this kind of research. We have a China Medical University as a company, a separate company. One of them actually is doing this kind of research product too, and also the Sinica, Director Chen, Director Huang, and also the Dean Cheng. And also we have our good friend, academician, this former president Hsing. He also is used to be on the advisory board of the Singapore A-Star advisory board, so they know each other very well. So without further ado, let's give a big applause to the former President of China Medical University, Professor Huang. Yeah, please. Yeah.
Jeffrey and Huang. This morning it's very exciting, and then I'm extremely honored to be able to be here. I'm not a scientist, but I think I probably need 2 hours to learn a lot of stuff. So in order to save time, because this morning we have 6 outstanding speakers to share the same notion of how to develop drug for neurodegenerative disease, and in particular to pay attention to the drug should be, as Jeffrey earlier mentioned, that the drug is unbelievable expensive. If I use a CAR T, at the time when CAR T just developed one person, it's $500,000 US, although it's become cheaper. So we need to develop drug, but the drug had to be affordable, sustainable, and should be inexpensive. So with that, then we have 6 speakers, and I was informed that Tom, you are the keynote, so we're going to start it first. Then after that, each one of you, we have 5 outstanding local speakers, and then each one no more than 10 minutes. So without further ado, so I would like to welcome Dr. Südhof, come to the podium to give his keynote, and do me a favor, another big applause. Remember yesterday we said we wanted him be able to come to Taiwan more often, right? Yes. 掌声不够大声啊！再大一点。
Do I need a microphone?
Yes.
Okay,
Happy
good.
better.
Thank you very much for this kind introduction. Thank you very much for coming here today. There was a bit of a mix-up in the preparation of my presentation here today, in that I prepared originally a presentation that would include both a general perspective as well as a description or an account of experiments we have been doing in our own lab. However, due to time constraints, I won't be able to give that talk. Instead, I will just provide some general perspective. If you're interested in the specific work we do in this field, I'm afraid today there's not the time to talk about this, unfortunately. But I think there are talks that I've given online, and I'm sure at some other occasion there might be an opportunity. So let me first, my disclosures, I've worked with a number of companies. Nothing I talk today about is directly related to my industry consulting. I have a number of roles in various companies. What I'm going to talk to you about is very, very general, because this was the initial introduction to my science talk, which I'm not going to give after all. And this introduction tries to outline where, from my personal perspective, we stand in the science community regarding Alzheimer's disease. What you see here is a picture of the first patient that Alois Alzheimer investigated. That was the indicator patient of the naming of the disease. This patient, Auguste Deta, had a familial form of Alzheimer's disease where a mutation in a presenilin gene causes a dominant form that induces the dementia symptoms already around the age of 50 and causes a rapid decline of the patient. As you all know, it starts with moderate short-term memory difficulties, referred to as mild cognitive impairment, and then progresses. The disease progresses towards eventually a complete loss of all brain function. It's a terrible disease, it's a chronic disease, and it progresses pretty much in a very canonical manner, depending on what the exact genetic background is. Alois Alzheimer already described the key features of the disease that we know of today, which are pathologically 3 features: tangles, plaques, and inflammation, that I will refer to later again. Specifically, the 3 key features that Alzheimer already described are shown here as the amyloid plaques, the fibrillary tangles, and the inflammatory response, activated microglia, astrogliosis. Those are the standard pathology features. In addition, there is a massive synapse loss that precedes, at least to some extent, neuronal cell death, which eventually kills the patient. There's been enormous progress in recent years, in the recent 2 decades, in the genetics of Alzheimer's disease. And the genetics that emerges is actually very, very similar to the genetics that is now observed in many other diseases of the brain, in that it shows a very interesting kind of pattern. What you see here is taken from a review that was published a couple of years ago, and that illustrates the genetic landscape. And what is important here is in this paper that this shows the frequency of the allele in the population. This shows the effect size. And you have on top here these three genes that produce disease 100% of the cases when they are mutated. APP and presenilins, the same cell biological pathway. These are the familial forms of Alzheimer's disease. Very rare, very tragic. On the other side, in addition to that, you have a rare mutation in one of these genes that's actually protective. Whenever the arrow is down, it means it's protective. Then you have here the very common variants that have tiny effect sizes, so tiny that they're unnoticeable and were only detected after analysis of hundreds of thousands of people. And these are very small effect sizes. They don't associate with any particular pathway that could be clearly enunciated and have been studied only occasionally. Even APP is there with a tiny effect size because of expression differences. These are the two extremes in the genetic landscape. And in fact, you will see a similar genetic landscape for other diseases, neurodegeneration, schizophrenia, autism. They all have a similar genetic landscape. There is no clear-cut, easy way of interpreting this genetics. But most importantly, what emerged from the genetics over the last decades are the ApoE genes. And ApoE genes, ApoE is a upper lipoprotein in the blood. I worked on ApoE and its receptors as a postdoc because it's very important for cholesterol metabolism. It transports upper lipoprotein, it's involved in the transport of cholesterol between the liver and the periphery. And so it was surprising that this gene turned out to be central for Alzheimer's disease, but it turns out that the brain also makes its own ApoE, independent of the liver, independent of the blood, and that the ApoE that is made in the liver is non-exchangeable with that of the circulation. And what you can see here is that ApoE is implicated in many different ways. There's first of all these alleles, which is very common, ApoE4. There's three different forms, 2, 3, 4, 1 doesn't exist. And the 3-4 combination is very common, huge effect size. This is the most important genetic variant. Then there's ApoE2-3, which is protective, it goes down. There's ApoE2-4 and 2-2, this is protective, this is deleterious. And there's obviously 4-4, which is still very frequent, has a huge effect size. The landscape, thus, that emerges is that the genes that are associated with Alzheimer's disease fall into three categories. The rare familial genes, the common genes that have very little effect size, and ApoE, and in addition another gene called TREM2 that's involved in inflammation. One general theme emerging from the many genetic hits in Alzheimer's disease is actually A-beta. And A-beta, as many of you probably know, is also the component, the central component of the plaques that are observed in the disease. A-beta emerges as a central component because these familial genes are associated with A-beta production. And because ApoE has been at least indirectly associated with A-beta, although that's connection is rather tenuous. The pathway that produces A-beta is shown here in the summary from Wikipedia, which I referred to yesterday as the source of all wisdom, which basically shows the APP molecule as a cell surface protein on the cell. It's on the neurons, on glia, on many other cells in the body. And the interesting thing about APP, whose function is still not quite clear, is that mutations in APP are one of the three causes of familial Alzheimer's disease. And APP is proteolized on the cell surface by two proteolytic enzymes, actually more than two, but two are shown here, so-called base and gamma secretase. Gamma secretase in turn has a subunit encoded by the presenilin genes, and these two presenilin genes are the other two genes where mutations cause familial Alzheimer's disease. So familial Alzheimer's disease genetics implicated already the same pathway as the morphological observation of A-beta in the plaques. And these are the enzymes that creates A-beta, and then the A-beta forms these plaques. A common model emerged. APP cleavage causes A-beta aggregates, A-beta release A-beta aggregates, and that causes neuronal cell death and inflammation, and most therapies as a result aim to remove beta amyloid A-beta because it may be toxic. This used to be the standard pathway until about 10 years ago or 15 years ago when people became more interested in inflammation. And since then, billions of dollars have been invested either in this pathway or in the inflammatory pathway. However, it has been relatively frustrating for the community, for the patients, and also for society. Because despite of billions of dollars of invested, these readouts have not been very promising. First, pharmaceutical companies developed BACE inhibitors that would stop this cleavage. However, the BACE inhibitors in clinical trials failed. In fact, if anything, they seemed to worsen the case for the patients. And so this approach had to be abandoned, which was explained at that time, at least, by the fact that BACE has also other substrates that are important. Then, A-beta antibodies as a passive immunotherapy emerged. Initially, they were not successful until more recently. This was a paper published about 2 years ago, actually pretty much exactly 2 years ago, January 2023, 2024, or 2023, 3 years ago. Okay, time flies. Unfortunately. Which was the first time any kind of therapy in patients had any positive effect. This paper is very, very interesting. It's very important and has been validated by other papers as well. And the difference between this study and previous studies that were not successful is that in this case the antibody preferentially reacted with aggregated A-beta, whereas in previous studies the antibodies would react also or even preferentially with free A-beta. And what you see here is that the therapy is extremely potent in that this antibody lecanemab, Eisai and Biogen, is very effective over 18 months in clearing the plaques in the patients. It is truly proof of concept in the sense that the antibody does clear the brain of the plaques. However, at the same time, the antibody had very little effect on the patient's development of the disease. Although this was used as the basis for approval in the United States and elsewhere of this antibody as a therapy, because there is a benefit. The patients did progress slower. The benefit is minuscule. If you look at it, the patients still cognitive performance score 14, others score 14. Now, you have thus the paradox that on the one hand the drug works perfectly well, on the other hand it's really not that good. Moreover, it is not that the treatment is too short. This is an 18 months treatment, and if you look at the last 30 months, then there's virtually no plaques left as shown here. The progression is just as bad. It doesn't actually get better. It's not like if you clear the plaques, the patients are finally slowing down the disease. It doesn't. So in other words, this approach at the same time was positive and negative. It was positive in that it definitely shows you can do it. It was positive in that there was a benefit for the patients. There's no question, this is a benefit. But it's negative in that despite clearing the plaques, the effect size is minuscule. It's really not that big. It doesn't seem to solve the problem at all. This is a benefit, but it's a small benefit. And in addition to that, there are huge side effects, a phenomenon called ARIA, which I won't go into, limits tremendously the usefulness, utility of this treatment. This particular paper and the follow-up papers were also interesting because they carefully analyzed biomarkers, both in the CSF and in the blood. I'm not going to go into that in detail for reasons of time. I just wanted to remark here that interestingly, in the CSF, the free A-beta with this therapy, at least A-beta 42, went up. For those of you who are not familiar with the biomarkers in Alzheimer's disease, it's classically the case that as the disease progresses, the free A-beta actually goes down. This seems to be counterintuitive, but it is explained by the fact that the A-beta plaques serve as a sink of the free peptide, and they sort of grab it all up, and they decrease the free concentration. So in that sense, the increase here and the fact that this didn't change for A-beta 40, another variant of the peptide, is positive because it reflects a reversal in some way of the process. But it's also important because it suggests that A-beta itself is not the toxic substance, because you wouldn't expect the patients to live better if it was the toxic substance. In addition to that, very importantly, CSF biomarkers for tau, which is the protein that composes the tangles in the disease in the brain, go down, which again is consistent with a positive effect. It also suggests that tau is downstream of A-beta plaques, not upstream. Phospho-tau. And plasma biomarkers, similar picture emerges from plasma biomarkers. They also show that the inflammatory responses go down. And at this point, especially phospho-tau is emerging as a major biomarker, both in plasma and the CSF. So the bottom line here is that the anti-A-beta antibody works extremely well because it works. It really does work. But and with also a decrease in inflammatory response. But the actual benefit to the patients is really minimal. Moreover, activating microglia also appears to be ineffective. In the clinical trial of a company that I called Electra, that I'm consulting for, which is a great company, the company found that the clinical trial of decreasing inflammatory responses by activating TREM2, where loss of function mutations cause Alzheimer's disease, had no positive effect on the patients whatsoever. It also caused ARIA, in fact, but it didn't help the patients either. So just going after the inflammatory responses doesn't work either. So my take on this, which I'm happy to discuss, even though I can't, for reasons of time, to discuss the actual science here, is that what is missing here is, in all these data, which are beautiful data, very important data, we don't actually understand what causes the disease. We don't understand why the patient's synapses die, why the patient's neurons die. We don't understand the role of ApoE really, whether it's related to cholesterol or to other functions that ApoE may have. In some, we don't understand what I call the cell biology of the disease. We don't understand what the basis is for the disease to develop. And because of that, in our own work, we have focused on trying to understand better the fundamentals of APP, A-beta, and ApoE at the most basic level, cell biological level, in order to gain insight into what may go on in this age-dependent neurodegenerative disease that will affect large percentages of our aging populations. So with this, I want to stop. I'm not going to talk about our own science here because of reasons of time, as I mentioned before. But I hope that this very general background, which is none of which is our own work, will give you some introduction to where our personal thinking is at present, how we believe this disease needs to be investigated in biomedical research, and why we believe that basic approaches to cell biology are the critical need for progress for a better understanding of the disease. Thank you very much.
First I'd like to thank Dr. Südhof that he actually cut down his own slide. So I actually count the time. Looks like we may still have a couple of minutes. That if somebody in the audience has an emerging question, only emerging question, you can ask more question, very concise. If not, then because later on we have to save 20 minutes or 30 minutes for discussion. So if audience, you will have a chance to ask questions for all the speakers. So our next speaker will be Dr. Wen-Chang, Superintendent Chung. He's a close friend of mine. We have 20 years friendship. And then he's very famous on, anyway, he's going to share with us using how to use smart science to cut the drug cost for the neurodegeneration disease. And here's one character, and I have to say, and Dr. Südhof doesn't have to know it. So I'm going to speak in Chinese. 全台湾的医院最赚钱的就是他了。What I'm saying is that the most profitable hospital in Taiwan was run by Superintendent Chung. Yeah.
It's my great honor to hear, not for the profit, but how to cut down the drug delivery. This is my topic. So far as we know, we have many types of treatment for the Alzheimer's disease or neurodegenerative disease. So far, antibody, small molecule, and antibody, and cell therapy, gene therapy, and etc. There are many kinds of the pros and cons. For the small molecule, the cost is very low. But for the antibody and the gene therapy, the cost is very high. But they are also have many advantages for the cure or the palliative treatment for each other. Then because the development the cost is very high, so if we want to develop a new drug, we must think again. First, we are find the new target, but the new target is very expensive to find. So if you have the position or the purpose of the new drug, I think by AI modify. I think the target the expense is maybe cut down. The next is how to monitor our treatment cost. We use PET scan, and it's very expensive. But we use spec below. And another, how to deny our trial members as small as possible. So it costs a lot. And we use IV intravenous usage or use oral or intranasal. Intranasal and oral is very feasible. Another, we are simplified our CMC the process and scaling scale up massively. The final we don't in Taiwan we need to know to ongoing to phase 3, phase 1 or phase 2. It will spin off our techniques transfer. I think this whole cost we can cut down our development cost. and especially for a small company, this is very important. So far is our experience. We use such as how to use AI to multiply our antibody or our molecular patterns. Such as this one, initially the molecular target antibody is very low affinity. Our AI modification, we get 10,000-fold affinity, binding affinity. And this AI is very important for development of our medication. How to sustainability of neurodegenerative drug. For the scientific sustainability, we want to develop one drug can treat multiple mechanisms, and one drug can treat multiple neurodegenerative disease. Such as I have an example, minimal peptide. It can treat Alzheimer's disease, it can treat Parkinson, it can treat ALS, and etc. This is very important. We can lower down our dependent free, such as one mechanism, many disease, and the small molecular and peptide is more cheaper. If we use gene therapy or use antibody, might be expensive. But in this field, I don't. My means is not to the antibody, but the antibody is very expensive. Our gene therapy is very expensive. But we also can going to do antibody treatment, etc. Another is use the drug is a platform is very better. One platform can develop many drugs and to treat many disease. This is very important. Such as we use modality IV antibody is another. The coherence to the patients is oral and the method is better. And in home use is better, not in the hospital. So I present three types of platform. One is small molecular, such as mini peptide and exosome. But it is a mini cost and immature and one country. There are three platforms can treat the neurodegenerative disease. As we know, the susceptibility high is a small molecular, exosome middle, and mitochondria immature. And scale up is very better. So if we want to develop a patient want a cheaper, effective, and easy to use, impossible. Okay. We ask AI for small molecular or not home. Can we use can we find this drug? Okay. From the AI, we have a small molecule. This small molecule is for originally is designed for detect the alpha-synuclein, detect Parkinson. So we modify this mini peptide from the detection to treatment drug. Okay, and we designed this PAA. PA is an abbreviation of Parkinson amyloid and Alzheimer's disease. This is a breaker to block the misfolding protein application. Here, block the 10 meta-sheet 5 together, and it can inhibit the tau, inhibit alpha-synuclein, inhibit DDR, the grade, and fell in the python and the other disease. Okay. So we designed a new type of the PA, cheaper, effective, and easy to use. We want to do PA is a variation of Parkinson's, Alzheimer, and amyloid. PAA. Is there? Okay, this is here. We can reduce the alpha-synuclein 5 years and better binding ability, the tau protein and amyloid beta, with very specific to penetrate the BBB in the brain. And initially the recovery, the running time in the low download, and for the Parkinson's disease also this is better PA, and the vehicle is very short in the running time. And the activity in PA is recovered. And the final, we can, if IV is not good size, but you can use oral or oral concentration also better, about 50% of uptake rate. Okay, this is the final. If we want to use a small molecule, this may you use an AI modified molecular pattern, and you can get a cost efficient and adherence to the patient in this method.
Thank you. So we also have time for, if you have an urgent question, raise your hand. One only. If not, that's fine. Then I hope that your what you call PPA is a general blocker for the beta sheet, right? So hopefully you're going to bring more money. Come to China Medical University. I'm the China Medical University faculty, so when you bring more money in, I'm always happy. Some of them I can use it. Okay. Anyway, our next speaker is also from China Medical University, from Dean of the School of Pharmacy. While we're setting up, let me just quickly say that Taiwan drug development has been ongoing for multiple decades. But those drugs which were developed in Taiwan and eventually all the way finished clinical trial and approved by whatever countries FDA-related organization. I think the first drug, Dr. Chen is in the team, that he has drug being approved to treat it with diabetes. I don't know how much money he made, but maybe you can ask him later on. But anyway, I hope that today's speech that he's going to share with us some small molecule and biological drug, right? And in the future, we're going to develop drug to cure neurodegenerative disease. Okay, Dr. Chen.
Yeah, thank you for former President Huang's introduction, and it's very honored to be here presenting my idea. Not actually the study I have in details, but since in response to the theme of the mini symposium today, it says cheaper and sustainable treatments. So my first slide says it all. Small molecule drug will be the way. Okay. And so one slide will be enough. However, I have 5 to 10 minutes, right? So this is the all the drugs approved by FDA CDER in the past 30 years. As you can see, the small molecule drugs indicated by the blue column here, still the mainstream drug discovery development. Okay, and although there are more biologics approved in recent years, and also noted that the more drug, actually more than first-in-class drug developed against to treat the rare disease. Okay, that's another opportunity for drug discovery and development. So, in terms of the CDER-approved drugs by modalities, they are small molecules, they are oligonucleotides, protein drugs, and as we learn, we know anatomy, we know physiology. So among those modalities, different modalities, the most accessible and can be delivered into the CNS brain system, that will be small molecules. Okay, so if we are talking about treating brain disease, neurodegeneration, let's go to small molecules. As we know, antibody drugs, there's only 0.1% and 0.2% in the circulation that can deliver circulating into the CNS. Okay. So small molecule drugs, or biologics, or ADC, or cell and gene therapeutics, which one is cheaper and which is better? Actually, because I'm in the drug discovery and development area, it doesn't matter whatever. It's small molecule, cell, gene therapy, as long as it works equal efficiently, they are all good medicines. And in that case, on that note, no matter how expensive, if the drug can treat a specific disease, it's a good drug. Okay? So I'm not saying small molecule will be only the way. So in comparison of small molecules and biologics, of course, small and large drug in terms of molecular weight. And because the molecule is small, it can penetrate through the lipid bilayer, so you can target molecules both inside or outside the cells. And if you are talking about penetrating blood-brain barrier, of course, small molecule, okay? But it has to have a very delicate design on the compound structure. So this is just a list of the comparison of small molecule and biologics as Superintendent Joe also presented. And what's the benefit about biologics and limitation? It's limited to the molecular target is limited to the extracellular molecular targets. However, if it can be internalized, it can be conjugated with toxin and internalized into the cells. Of course, it can be used as a good drug as well. And with antibody, it has more specific recognizing target molecule in comparison with small molecules. Okay. And without very little known about neurologic disease in terms of mechanism and etiology, and this is what happened. People have longed for effective treatments for neurologic disease. So this company has a stock on market in the US market, and its target is actually a TCM only, traditional Chinese medicine, and a claim to treat ADHD and ASD. However, it's still in the clinical trial. But in the market, the response so dramatically, it increased 100%, that's 804 in market price. And then after the surge, it returned to about 17.4 as its first initial IPO price. So the company is selling a dream, or it can be effective TCM formula at the end, remain to be seen. And this is the very first CAR T-cell therapy approved by US FDA and covered by our national health insurance. it costs about 475,000. That is expensive. However, it's also covered by our national health insurance. Okay, and compared to Kimbraya, compared to this on the list, the price is these are even more expensive than the Kimbraya. And among those 10, only 2 covered by our national health insurance. So, how many others can we cover by national health insurance? Okay, they are too expensive, right? And just to mention the price about this first gene therapy for hemophilia B, hemophilia A, they are all around 2.9, 3.5 million US dollars. So still too expensive. And Pfizer just announced last year they give up on their cell and gene therapeutics. There are no more pipelines on their for the cell and gene drug discovery. However, as I mentioned earlier, no matter the small molecule or cell gene or antibody drugs, as long as they are equally effective and treating the same disease, same patient population, they have equal market value at the end when they launch on the market. Okay, so eventually maybe small molecule is cheaper, biologics is more expensive, but at the end they are all the same in terms of market value. So this is CDER, FDA CDER approved by therapeutic area. Of course, oncology is still the main focus. So there are more drugs in discovery and development area and finally approved by FDA. And neurologic disease is a second focus of the area. And this is year 2024. Also, 5-year average neurological disease is still the focus of the year. So, if we are talking about disease, about neurogenic disease like neurodegenerative disease like Alzheimer's disease, and as Professor Südhof just mentioned, we need to have more study. We need deep molecular cell biology studies to identify druggable molecular targets. So there are a lot of works for us biologists to do, and we need mechanism of action that correlate the etiology of the neuronal disease, and the etiology has to have very specific identified druggable molecular targets. So just to share with you that most of us probably we are working on at this area to identify molecular targets and to validate the target in preclinical and also clinical settings. And after that, there are actually a lot of work to do in the discovery area and also preclinical and clinical development area. So eventually, it's on the market if everything goes smoothly, right? And it takes multidisciplinary scientists to work together, and the collaboration has to be very close and intimate. So it's not that easy to say that we can do drug discovery. This is a case that we have developed a drug on the market last year, approved in mainland China, and it's pusoglyptin. It's a DPP-4 dipeptidyl peptidase 4 inhibitor, and it took about close to 20 years from the very beginning to the end. Okay, and we did not spend a lot, as you can trust me that we spend very little in comparison to the money amount that you have to spend for one case of drug discovery and development like this in the US. Okay, it's about 1/4 of the cost compared to the US. Okay, so this is probably my last slide. So if you identify any unmet medical needs like neurologic disease, it's more important, Alzheimer's disease, let's join together to have some novel drug discovered and developed for the benefit of patient, not for our own good. Okay, that would be okay. Yes, thank you.
Thanks, Chen. I think the next speaker that's prepared. But any question? We still have some time at the end and ask a question. But Dr. Chen just pointed out a very interesting and I think a very critical question. That is drug cost. $3 million US dollars. Taiwan Taipei 多少钱? I'm sorry, I asked them to translate to $3 million US dollars is how much NT. Too expensive. So when I saw that number, I thought, well, maybe in the future for the drug development, well, those expensive drugs, maybe just like you buy a house, you have to pay one mortgage first, right? Well, anyway, that's not what we'll discuss here. But if there's no question, we thank Dr. Chen. And then this is a very important question for small molecule against large molecule, and then the cost. And he's mentioned that all these are drug development, but fundamentally, the question, yeah, go ahead.
I totally agree that 3.5 million is a lot of money.
Yeah.
But it's more a question of how you finance drug treatments. Because if you think, for example, of sickle cell or hemophilia, the annual costs of treatments go into the hundreds of thousands. So if you can stop the disease with one gene therapy at a young age, that's totally cost effective.
Yeah, true. In particular for this case, yeah. So there are a lot of another dimension of regarding the cost and then the prevention for the nation to expend the money. And since you mentioned that, people is joking that well, maybe in the future it's not a joking. If I need to use a $1 million drug, but I'm not sure I'm going to apply. So just like a mortgage, I pay the first down payment. If treatment work, then I'll pay the down payment in 30 years. But if I die, then I only pay down payment. I don't pay the rest. Well, anyway. So, and then it's an interesting point. So, in addition to the drug development, I think it's important to understand, as Dr. Südhof earlier mentioned, that to understand from the molecular cell biology point of view, the fundamental science about the disease, and then from there you may be able to develop the drug. So our next speaker is from Academia Sinica, Cheng, Director Chen from our Institute of Biomedical Science, and then she's going to share with us her basic research, and then from the mechanism study to identify potential drug. And while she's setting up, hey, 他是长官啊，hey, 长官啊 ，she's the officer. She's back there academic now, but she used to be running the National Science Technology Council. So at that time, all our grant to be approved had to be. I have been always very nice to her. Even today, I'm very nice to her. Yeah, anyway.
Yeah, good morning. Thank you for the kind words. I've been in anesthesia maybe 2 years ago when I came back to Academia Sinica. My name is Chen. It's truly a great honor to be here. I can tell you, my dream is to, before I retire, there could be some therapeutic possibility for Alzheimer's disease. Now, thanks to Dr. Südhof, now we're all very clear that Alzheimer's disease has at least two major features: amyloid that trigger and that reduce synapse loss, and also hyperphosphorylation tau that would actually transmit from one neuron to another and cause AD progression. Now, I would tell you there's another feature that is also super important that people have reported, which is energy failure. Now, just imagine that as electricity of this building. Now, if that is a problem, can we improve Alzheimer's disease by restoring energy? That's the question that prompted my group to actually look into this question. That lead to another molecule, adenosine. I trust that all of you in the audience know this. Adenosine is an important neuromodulator that links neural activity and energy metabolism. Now, through purine metabolism, people already know that adenosine is linked to energy productions, neurotransmitter synthesis, and DNA, RNA synthesis, signal transductions. So you can tell that this small molecule actually sits in the intersection of energy, synaptic transmission, and also signaling. Now, this very important homeostasis actually is disturbed in Alzheimer's disease. Let me tell you why. Now, we all know that in Alzheimer's disease, release of glutamate from the presynaptic terminal here actually was enhanced. So there would be tremendous accumulations of glutamate being accumulated in the synapse that activate an DA receptor, NPI receptor, eventually opens up the channel, turn elevated intracellular calcium, and probably also cause mitochondria stresses, eventually cause excitotoxicity. Now, this is a general knowledge. However, this intracellular increase of calcium also leads to a very important discovery reported a few years ago that this calcium also triggers in an activity-dependent release of adenosine, which means that adenosine released from intracellular to extracellular. So what would that happen? When that happens, it would cause nucleoside dyshomeostasis. Now, I'm not telling you just a hypothesis. Actually, in human Alzheimer's brain, what you will find is that in AD's brain, the intracellular adenosine level is much lower, particularly, very interestingly, particularly in early and medium stage, which means that this lack of adenosine might actually contribute to the synaptic vulnerability. This might occur way before excessive neuronal death. Now, if this is a problem, can we solve it? Let me bring you to another important molecule called ENT1. Now, this molecule actually sits on the plasma membrane in synapse. It governs the transport bidirectionally of adenosine. Now, if for AD, the adenosine defects is from the intracellular outside, now can we block this molecule? So in the past 10 years, we established, it took us quite a few years to establish a compound called J4. This is an ENT1 inhibitor. It is orally active. So we test this hypothesis. Now, what we had done is to look for mouse models of Alzheimer's disease, tau-22, and we look into the brain. We found indeed in the brain, if you compare to the red bar to the black bar, you found a reduction of adenosine in the brain. Now, if we treat these animals with J4, shown in green here, we saw a slight increase in adenosine. However, most importantly, that it being converted into ATP. So the J4 treatment actually increased the synapse ATP, not necessarily synapse, but the brain ATP level. So what happened is that we believe governing adenosine homeostasis is one effective and possible way that could actually control energy metabolism. Now, this beneficial effect not only limit to ATP level. This is a summary slide. Let me tell you that treatment with J4, this small molecule, orally active, that it improved memory and cognitive function, and reduce accumulations of amyloid and p-tau, it reduces the impaired mitochondria, reduce the elevated brain oxidative stress, even restore the impaired glucose metabolism. We also found that it improved the AD-associated sleep disruption. Now, you would probably feel really weird that how come one drug would cause so many beneficial effects? Now, let me tell you our thought. We believe that it is because all these diverse effects is because in the brain, most of our function, including synaptic transmission, actually require lots of energy. So even a modest deficit in ATP availability would actually disable many biological function in the brain. So if we provide energy to actually restore a permissive energy state, that would allow these functions to function properly, then maybe we'll be able to improve the brain function as a whole. Now, we have tested this molecule in the brain in 4 different AD mouse models. It also received FDA and TFDA approval and is currently in phase 1 in Taiwan. Now, besides that, I would also like to emphasize one very interesting point is the importance of microglia. Now, as Dr. Südhof had mentioned, this is a very important cell type that would contribute to Alzheimer's disease. Now, many studies already mentioned that TREM2, ApoE, lipid, cholesterol metabolism are important. I will bring one additional molecule to you that called galectin-3. Now, this is a molecule. It's very interesting. In astrocyte, using single cell, single nuclear RNA-seq, it could actually separate microglia into 12 clusters. And this galectin-3, it is a molecule that binds to beta galactocyte. It is highly elevated in cluster 9, which is a disease-associated cluster. Now, upon stresses, galectin-3 will be elevated, and in the brain, that inside the cell, that it would actually sense lysosome damage. Now, in AD, it is even more interesting because what it would do is that it could be galectin-3 could be co-released with phosphorylated tau protein, and what happened is that it could in the EV, and galectin-3 will facilitate the aggregations of p-tau transmitted to another neuron and facilitating further enhance AD progression. So that is why that when we block galectin-3, it also improves Alzheimer's disease and also another Huntington's disease. Now this is my last slide. Now with all the data that I show you briefly and our 2 possible mechanisms, the energy mechanism and also neuroinflammation mediated by galectin-3, together with many other really very important mechanisms, we would like to think that maybe new targets, new strategy, and even combination treatment could be what we needed today for such a complex disease. So with that, I would like to be able to further discuss with you at the end, and thank you for this opportunity.
Chen, from the basic science study, he identified J4, and then even in fact those very common molecule like ATP or adenosine, which is we all supposed to know from biochemistry, and plus galectin-3 was very interesting. Any particular question? If not, thank Dr. Chen. Yeah. And then our next one is neural drug. Behavior is also very important. We all behave, right? So I hope that I behave well. So anyway, Dr. Liao.
I didn't thought that I will have a much more time than I thought, because every speaker here saved a lot of time for me. But I won't extend my talk too much, because I would like to leave the more time, if possible, leave it for Q&A session. Thank Professor Huang, and it is an honor and a pleasure for me being invited here with this wonderful panel discussion. Thanks President Tsai for organizing this wonderful Nobel Laureate Forum, two-day meeting, which are very exciting. My student and I has been very exciting lately for this event. And especially we have all distinguished panelists, especially with Professor Thomas Südhof, coming a long way from Stanford University, California, United States. I hope some of the students here may go to Stanford University in the near future for their advanced study or even a short-term exchange. As mentioned by Professor Südhof yesterday, more academic exchange, more scientific communication, and I believe leading for research excellence in the future. And so that's how you can see how I'm so excited right now. Okay, and because I have more time than I thought, I would like to maybe I would like to carefully present my talk by watching my screen, not missing any words for my. Sorry. So I like to switch the gear a little bit from biomedical science to behavioral science a little bit. So my title is From Mechanism to Meaning. And I would like to address how behavioral neuroscience could be a functional bridge in sustainable drug development and for neurodegenerative disorder, and perhaps neuropsychiatric disorder too. And I will explain that. Okay. Before diving into my topic, I would like to briefly highlight the interdisciplinary landscape as represented in this panel. Today we have expertise from clinical neurosurgeon and biochemistry and pharmaceutics and cellular and molecular neurosciences. For my own work, I'm focused on behavior, linking neural circuit and observable function. This multiple level perspective should remind us that understanding brain disorder requires collaboration across every layer. Just mentioned by other speaker, namely from molecular to behavior. Furthermore, in sustainable drug development, this connection allows us to translate mechanism into outcome that truly matter. Okay, let me explain why behavior is important in drug development. In behavioral neuroscience is not option in brain drug discovery. indeed, it is essential, as brought up by other panelists earlier. And often, the drug development for CNS disorder require more than molecular or imaging marker. In fact, many neuropharmacological mechanisms are best reviewed by using animal behavior model. To evaluate functional recovery behavior endpoint must be included alongside molecular and imaging marker. Shortly speaking, neuroscience needs behavior. And as I showed one special issue published years ago from Neuron. So I would like to explain why behavior as a readout in quantifying functions. In fact, behavioral tests based on conditioning paradigm reveal meaningful drive on distinct component of cognitive and emotional regulation. In contrast, unlike the reflexive behavior such as locomotor activity or simply the sensor responses that the conditioned behavior model could capture goal-directed learning, providing essential insight into high-order brain function as reviewed for the psychiatry and the neurodegenerative disorder. Importantly, research in psychopharmacology will allow us to link receptor action to functional outcome. Let me give you an example, which you may not be familiar with, the so-called DRL, differential reinforcement of low-rate behavior. As shown in this diagram, you can see that two types of operant conditioned behavior. On the right panel, showing that FR1 test versus on the left side, the DRL 10-second versus. Although these two kind of behavioral tests all belong to operant conditioned paradigm, however, they have difference in terms of response rate. One is high response rate. The other, DIL, is in low response rate, as the term showing that its character, behavioral characters. Just give you an example that how fast rats trained in the FR task. For instance, FR20, the rats can press the lever or the operandum maneuver in more than 1,000 times per 30 minutes. In contrast, if the rat trained in a DRL 10-second task, it would only generate about 150 lever presses per 30 minutes. So they are quite different. And why is that? Because especially in the DRL test, the rats need to learn two components. One is behavior inhibition. The other is kind of interval timing. In this case, 10 seconds as a criterion interval. And then the rat has to withhold mostly we use the male rat, so I hope it's a response with impulsive control in terms of behavior inhibition, and try not to press the lever until the criterion interval, for instance, 10 seconds elapse. Otherwise, this rat would not get any reinforcer or reward. So in this condition, a rat really can learn how to withhold its responses and then waiting for a certain time, like 10 seconds. And in this slide, I would like to explain more about the behavior measurement under DIL 10-second task. We collect the data from the behavior responses made by the rat, and as you can see the panel on your left, each color dot meaning represent each lever press or each response in seconds. Okay, and so, but between the responses, we measure the inter-response time, initialized, abbreviated by IRT. We collect this IRT data and then put into a distribution curve and turn out the IRT distribution something like the curve shown on the right side. And from this IRT distribution, we categorize IRT by IRT second beam from 1 second up to 20 or more seconds, and then we calculate what's the frequency in each beam. So as you can see from this IRT curve, represented by a red, kind of a train, well-trained in this DIL 10-second, by showing that the peak located on axis about 10 seconds, meaning that the rats kind of learn how to wait for 10 seconds in order to get a reinforcer. And then through this IRT distribution, not only the peak time or the peak rate, as I just showed you the peak time, and then you can see how high the peak rate indicated by y-axis as peak rate. And then peak time and peak rate respectively can represent the timing process and the motivation. And other than that, we can calculate all kinds of, four kinds of responses identified by the total responses. And for the responses made within 10 seconds, those are categorized by non-repose responses. In contrast, the responses made above the IRT over 10 seconds, those are the reinforced responses. So meaning that we have a multiple behavior measure at the same time, we can use to evaluate how the rats responds. And recently, the DIL 10-second or DIL test has been increasingly used to evaluate the neurobiology or psychopharmacology of impulsive action. And I believe impulsive action is highly relevant to psychiatry and neurological disorders as well. And just give you two pieces of the data we recently published. One is that we use the DIL test and then try to understand what the neural substrate could be underlying this impulsive action test. We use rats and then train them over 14 days, and then as you can see from the upper the panel, the left figures, then there are two groups can be separated. One is high impulsive, and the other is low impulsive groups, represented by red and blue colors. And from the bottom left, and showing that the IRT distribution curve observed from the first day and 7th and then 14 days. You can see the IRT distribution gradually shape up on the 14 days. And those quantitative measures underline the curve, as you can see from that. And we can easily tell from there are significant group difference between group difference. And then we look into how brain glutamate system can be involved in this type of behavioral phenotype. And we use the MRS, magnetic resonance microscopy, to detect the glutamate signal. There is a difference between high and low impulsive group. As you can see that in the nuclear compass initiated by NAC. And then further, we use the Western blot to differentiate what's the NMDA receptor subunit could be differentiated in these two groups. And then we found that there is a significant difference on N1 subunit of NMDA receptor between these two groups in dorsal striatum and the nuclear compass. Not only the NMDA receptor, but also the metabotropic receptor. We recently found that they also especially the group 1 metabotropic glutamate receptor also involved in this type of time-based impulsive action measured by DRL test. So as you can see, I just go through on the right panel. You can see I point out that the rat treated with 10 milligram per kilogram of NTEP, and their responses drop, especially on the non-reinforced responses, especially located in 8, 9, and 10 seconds. Those data suggest that this drug has some improvement for the response inhibition, meaning that the rats may have a better impulsive control in this condition. So just move on to thinking about how behavior can be translated into our functional outcome by using the animal model. So I just gave you an example, especially using the DRL in terms of impulsive action. And in this context, as mentioned earlier, many CNS drug value rise from poor translation between molecular target or functional outcome at the behavior level. So it is important integrating this behavior model into early phase drug trials can maybe help to enhance target validation, reduce development risk, and support a precision medicine. This will be my last slide. So I would like to just address that how important the behavioral neuroscience can be used in understanding the mechanism of the brain disorder from molecular to behavioral level. As we know that the multi-level framework enables the development of mechanism-driven, functionally validated drug strategy. At last, precision medicine ground in such integration improve efficacy, reduce off-target risk, and enhance long-term sustainability in drug development. Thank you for your attention.
Chairman Liao, then I think that I would like to take this opportunity to thank all this morning all 6 speakers for outstanding talk, and also you guys are great, good citizen, keep in time. So we have enough time. So I think the coming section, if I understand correctly, all everybody to come to the audiences. So now we'll transfer to all you guys. Don't leave. Scared? That it's time. It's your time for you to ask questions. But so should we invite? Should we Dr. Südhof, and then everybody, all the speakers, could you please come sit down? Do you need a 5-minute break, or it's okay? Do you need a 5-minute break? Yeah, okay. Then let's sit here. And then while everybody try to sit down, I'll take this opportunity to thank that our host institution, the Asian University President Tsai, and also our International Peace Foundation head, my friend. Oh, you were there. I thought you were there. I'm sorry. Okay, Dr. I usually call you Wolf. And because your last name is more difficult to pronounce. Your first name is easier. But thanks for International Peace Foundation and Asian University joint effort to invite Dr. Südhof to come this morning. I don't know how you guys feel. I don't know anything about neuroscience, but these 2 hours, it's like multiple year learning for me to learn neuroscience. Okay, so with that in mind, that I think we can start it for the audience question. And I remember yesterday, a question from audience. No.
It's really open.
Okay, it's open. So it's your time. So I'll give you the time to write down your question and then prepare. You're going to ask a question. So to save time, anybody have a question now? You can raise hand out. Then you talk first. Okay, then one. Could you do me a favor? Identify yourself. In particular, if you are a student or postdoc fellow, then speak very loud.
Going to Stanford University.
Oh yeah. There's one question there. You probably need to use a microphone. Do you need?
Microphone there.
Thank you, Dr. Huang, President Tsai, Dr. Südhof, and all the panelists. It's my honor to be here, and my name is Li Jun, a senior student from the Department of Psychology here at the Asia University. And I would like to ask a question to Dr. Südhof. My question is that your work has defined the molecular machinery of neurotransmitter release. Early in your career, you focus on purifying synaptic protein using biochemical method. And how did those early challenges shape your view of synaptic complexity? And today, what big questions still drive your research on synapse formation and self-adhesion molecules such as neurexins? Thank you.
Yeah, so that's a question that obviously has nothing to do with Alzheimer's disease, very little, but I'm happy to address it. Unfortunately, I've been in science for quite a long time, and it's a reflection of my age. When I started, there was no genome sequence, there was nothing available. Protein sequencing had just been recognized by a Nobel Prize. It was a different time. Interestingly, before this event here, I had the opportunity to go through an exhibition that is in this very museum. And that exhibition emphasized the changing nature of life, of our world. Similarly, science is changing continuously. It has changed over the 4 decades I have been basically a professor, which is a long time. I was a professor very young. And so, as the changes happened, so has our research and our view of the brain. When I started science, there was, as I mentioned before, no idea what a gene would look like. We didn't know there were introns and exons. We didn't know what proteins were like. We had no idea of any of this. It's hard to imagine. There were no computers either, so just no personal computers. Okay, my thesis was written on a typewriter, which taught me how to type. It was very useful. So the short and long of it is that as our science changed over the decades, so has my view of the brain, and so have the approaches that we are using. In addition to that, so has my interest. I used to be primarily interested in membrane traffic and in how neurotransmitters are released. I don't work on this anymore. Other people do, and they do much better work than I could ever do. I have changed, and I'm much more interested now in understanding the logic that guides the construction of the brain, that guides the emergence of circuits. I'm interested in that because I believe that we need to understand that logic in order to understand behaviors, in order to understand the complexity of the brain, that we can't just describe connectomics, that we actually need to understand how the grammar of the brain, if you want to call it this way, because the connectomics are just going to be like language. The possibilities for connections are infinite, but the grammar is always going to be the same. I don't know if that answers your question, but one of the, I think, key features of science everywhere is that we need to change continuously. We need to adapt, and we need to try to find ways of making use of the technological advances that we have the opportunity to use. The technology has changed so dramatically over the last years that I think that many in science are not quite maybe aware enough of how much that has actually changed how we think and how we approach an understanding of the natural world. And that is one of the biggest issues, because that also translates into, for example, into problems in drug development. It's today's subject. As really we live in a different world now than, for example, only 10 years ago.
Well, thank you. If you have a question, please raise your hand. And just in response, I'm waiting for you guys to raise your hand, so I just free all the time. Oh, okay, the lady there. Yeah. I think you will say yourself. Thank you.
Thank you to all the panelists for your inspiring talk. I'm Edith, a double major senior student from HR University in psychology and biochemistry. And I would like to direct my question to Dr. Südhof and Professor Chen. So my question is, from your experience as a neuroscientist, what do you see as the biggest challenge in translating molecular and cellular neurobiology findings into clinical therapies for neurodegenerative disease today? Thank you.
Okay, thank you. So, lady first, I understand.
Okay, thank you. Thank you for the question. This is really a very important one. As Dr. Südhof had mentioned, the science, the approach, the knowledge has changed so much. At this time, I personally think that cell-cell interactions, because we're not looking at one single cell, we're looking at multiple cells. They are neuron, they are also glia, astrocyte, microglia, oligodendrocyte. How do we use one treatment or combination treatment to actually control effectively? So that is the thing that I think at this time that is most challenging to me. And I wonder, Dr.
Yes, this is an important question, and I think that my personal view is that people are way too quickly going into translation. There's a tremendous pressure to translate from the community. In addition, individual scientists feel often that they should put translation first because that's what the funders want, what everybody wants. Then it is afterwards, when the clinical trials fail, then it is said that, well, it didn't quite translate well, the animal models are not good enough, well, whatever. And the fact is that many of the trials, I think, that are being done should never have been done in the first place, because the clinical science, the preclinical studies weren't there. So in every country, increasingly, I don't know about Taiwan, but in every country, government leaders feel that there need to be more clinical trials, we need to document, we need to apply, and so on, right? It only leads to increased failures. Many of the clinical trials, for example, Alzheimer's disease that have been done, I think are a total waste of money and time. And they direct or distract, they misdirect funding because clinical trials are insanely expensive. There are, so in drug companies, clinical trials usually account for more than 90% of the total budget. Often more than 95% of the total budget. So I think we need to have a culture in science and biomedical research where people are more careful about what they need to translate before they translate, and that we as scientists, as preclinical scientists, are often also too ready to give in because we want to get the funding, we want to get the money, we want to basically be seen as being good scientists. We are too ready to give in from the pressures from patients and from governments to translate stuff that isn't really quite there yet. It is actually more of a bet than it is a rational behavior. And if that then leads to failures, then comes the complaint that the science is not good or whatever, whereas the real problem is the juncture between translation and preclinical research. I'm not sure everybody likes this kind of view, but I think it's important to keep in mind that it's often not the science that's the problem.
Okay, thanks. More question? Okay, there's one lady over there. Yeah.
You want to go first? Yeah? Okay, so I guess some students will ask later. I would like to follow up on the combination treatment and even lifestyle intervention, as Professor Huang mentioned. So my understanding is Alzheimer's disease is really multifactorial, and really all the lifestyle, like diet, and also behavior, like the social interactions, and also like energy deficits. There are so many pillars being affected. So I guess it's not that easy to design a clinical trial to really have very good, like convincing result. And also there are so many confounding factors which may be influencing the result. So my question is about whether there are any suggestions on that front of making those combinations really readable and have a clear outcome that can really convince the society, and also building better care systems, like in hospital, long-term care centers, and even in the earlier stage, like in mild cognitive impairments.
Thank you. So your question is combination. So any specific person would like to answer from the panel?
So yeah, because the combination actually goes through like Professor Huang, Professor Cheng, and also of course others.
Professor Chen. Chen is this one.
Yes.
Is that? Anyone of you want to volunteer? Go first. No. Well, anyway.
I'll start really quickly.
Actually, I totally agree with you. And many senior professors told me that it's very difficult to do clinical trial with combination trial. Okay. However, if you look at the biology behind, I personally do not believe that you could actually treat one type of cell and actually get very effective treatments. So since last year, 2025, Alzheimer's disease drug development platform, they also start advocating that combination treatment might be needed. But what you just mentioned is really important. How do we, in the system, to do that? That to me remains to be a question. Yeah, thank you.
So.
I think I totally agree with your point. Combination is really important because if you look at the AD, it's not only neuronal cell death, but also neuroinflammation happens there. So I don't believe one drug can treat everything. Certainly combination is really, really important. Just looking at my own study, I believe not only one bacteria can do everything. Instead, maybe combination of different several type of probiotics or postbiotics can do the thing.
Okay, so with that, I want to add that, Tom, do you have anything to add? Okay, so I would like to say from the cancer point of view, combination therapy is standard now. And when you want to do this combination therapy, it is important to have a detailed mechanism. As Dr. Südhof earlier mentioned, well, clinical trial is certainly important. We should all move to clinical trial, otherwise there's no drug, right? However, when you move to clinical trial, you talk about billions of dollars, right? But without complete mechanism, you go ahead to run into the clinical trial, you fail, and with a very trivial reason. And in the cancer area, that has been occurred very, very often. For example, a lot of drug companies do a clinical trial, they use the skin mice to show how tumor spread. But when you use the immunocompetent mice, just like a human being, you have the immune response. It doesn't work. But there are a lot of these kind of examples. So with that, I'd like to emphasize what Dr. Südhof and earlier Huang also mentioned about mechanism is important. You need to know why, and therefore design the specific therapy. And I think a neurodegeneration disease is coming. This is certainly a big issue in the human being. And then I'm glad that we have this opportunity here to discuss it. Any more questions? Or okay, so we have two more here. You go first, then you, and then you have comment? Yeah. So before that.
There are different ways to do the research. One is for pure research. You can use the combination therapy because the cost is low. If we want to develop a new drug development, you need to simplify your target, not use the combination therapy. In antibody therapy, this is for amyloid level. You can check amyloid level accumulation in the brain and include the criteria is very simple. If you can not use the very conventional therapy, use the antibody or use the lifespan adjustment. This is too complicated, and that is very expensive for drug development. This is my talk.
Okay. Take the Dr. John's input. So you go first, okay. Then you.
Thank you for Dr. Südhof for your inspiration. And I am coming from the optometric department, and we appreciate that the insight of you that whether your related study could be applied to the neurosignal transaction in the ocular retinal cells. Thank you.
So could you repeat that question again? And then
I'd like to know
the real
neurosignal transaction, the theory could be applied to the in the eye, the retinal cells. And we think that the retinal cell is a part of the nerve cells. Okay. So I would like to know that this study could be applied to the especially for the degenerative retinal disease. Thank you.
So you're talking about retinal eye disease. Okay.
Yeah.
Any question? Anybody want to respond? You beat me now. I'm a cancer biologist. I only know retinoblastoma is one of the tumor suppressor gene, but I don't know retinoblastoma relate to the neuroscience. So unless we rely on them, we have a limited amount of time. So if there's no specific answer for your question, then maybe later on we can discuss that. There's another lady over there, question there, and then one more over there. Yeah, okay? Go ahead. I want the same time. Sorry that, but we can talk about retinoblastoma in the future. I thought there's a lady over here. That's fine, yeah.
Okay, thank you for all panelists giving a thought-provoking talk. And I'm Eva, a sophomore major in psychology from Asia University. So my question may go for Professor Wang. Okay, my question is that in systems like the gut-brain axis, it can be hard to link what's happening in the body to how people actually behave. So do you think using AI or computer models could help us to understand that connection better? Thank you.
Okay, so Dr. Wang.
Yeah, I think certainly AI is really powerful. I mean, at this stage now, AI is everything. You can use AI to characterize maybe from a huge population of people. You collect very huge amount of data. You can analyze what material composition in different populations, right? That would be very helpful to identify disease correlation with maybe material composition as well as the metabolite that's secreted. I think it's the future.
Okay, so there's one more question here, right? Yeah, go ahead.
Thanks for all your sharing. And I'm from National Taiwan University. As some of your lectures mentioned before, the study fund is very expensive. And why do you believe it? Your candidate and platform has the potential to cure the neurodegenerative or create some cure medicine to these patients. Thanks.
So I only hear you say expensive. Yeah. Okay, anybody want to answer? Thank you.
This is a very good question. I could tell you that when we started the clinical trial, before that, we actually tried 4 different AD mouse models. They all have multiple mechanisms. So those data convince us, preclinical data, that this is possible. But again, this is a very difficult decision. I think you are absolutely right. We should have very confidence before we actually start doing that.
Yeah, we never give up hopes, especially for patients. Even though, as we mentioned today, medicine, some are very expensive, and although I was selling my idea about cheaper medicines. However, if the medicine is at its price, at its cost, that can match the patient's needs, I think we should go for it. Okay, that's my idea.
Okay, you satisfied? From expensive to not expensive? Are you okay? Okay, more question? Oh, so many now. Okay. The lady and the other lady behind, then there's one more there. Yeah.
Okay, thank you, professors, for your inspiring presentation. I'm an undergraduate student in psychology, and my question is, since patients with neurodegenerative diseases often have both thinking and mood problems, if combining lifestyle-based approaches such as changes in dietary intake and gut microbiota with medications that target key proteins, it be better for long-term treatment? Thank you.
A question about microbiota? Yeah.
Well, I think because it's simple, right? If you can just make your diet in a good way, that could make your body become healthier. It's definitely the way to go, right? You don't need any medication. But in another word, if you think diet is not sufficient, certainly combination of diet and medication is necessary. I hope I answered your question.
There's one more question behind here. But to save time, that you could have a microphone. The one here, you already microphone, one I go first.
Hello, so I'm Marco, currently a high school senior. I actually have like 4 questions, but I'll minimize it to 1.
Sure.
Okay. So I want to ask Professor Südhof. I really like how you kind of deduct from a lot of potential pathways that we can go through. And is it possible to map a causal pathway through like familial AD risks and also like sporadic AD risks. For example, like AD due to like A beta, then like microglia, and like let's say to synaptic like dysfunction. Is there and do you see like if let's say is there anything like in the Südhof lab like working on that currently? It's like
Anybody understand the question? Those who understand the question, go first. Hey, I'm not a neuroscientist. I have no idea what.
So as far as I understood, you're asking me, right?
Yes.
Okay. So yesterday's discussion asked a question from a student, I believe, or maybe it was a faculty, I'm sorry, how to, at the same time, be conversant with the details in the expanding amount of information in science and have a broad overview. In Alzheimer's disease, the number of studies that are being published are just impossible to read. There is so much, it is so broad, that anybody who tries to understand what's going on is facing a major challenge. As a result, people tend to focus, sometimes a bit narrowly, on their various areas and to overemphasize them. And that is in many ways necessary because each individual lab can only work on one thing. You can't work on everything in a field, that's impossible. But at the same time, the danger is that you become biased in terms of your thinking. Being aware of this, what we try to do is we try to follow, in general, what the field is doing. But in particular, then choose one area that we believe has potentially the best chance of providing some deeper insight into the disease, which in our case is focused on A beta and ApoE because of the genetic guidance here, that those are the major pieces. That doesn't mean that the other pieces might not also be important or might not be more important, but in our own work, that's what we do at this point. I think the challenge in Alzheimer's disease is really that in any kind of conference you go in, even here to some extent, people have their own view, and that's what they do. And we need more, not interdisciplinary research, but rather more interperspective discussions where we basically think about the entire landscape and evaluate everything that's in a landscape and then try to see, reserve judgment. So for example, in microbiome, there must be thousands of papers on Alzheimer's disease microbiome. And the question arises when you look at these papers, which one do you actually believe and which ones have borne out, and what does common sense tell you about the microbiome in the disease? For example, is there any epidemiological evidence that people who have gotten a lot of antibiotic treatments in their life have a higher or lower incidence? Okay, because that would dramatically change the microbiome. These kinds of questions. I often find that in science, people don't approach questions with common sense. So there's a lot of common sense sort of tests that you can apply when you look at the overall landscape. And it would be very helpful for the field if we had more of these types of discussions. I don't know if that answers the question.
Yeah, I think that answers my question. Thank you.
Thank you. Then there's one more question there. It's getting late now, so go ahead.
Thank you, Professor Huang. I'm so pleased to be here and meet distinguished professors. I'm Yujie, a psychology student here at Taichung University. My question is for Dr. Tamasudo. Thank you for your inspiring talk. Your research indicates that mutation in synaptic molecules, such as neurexins and neuraligens, are linked to neurodegenerative diseases. Do you think Similar synaptic dysfunction also plays a role in psychiatric disorders such as autism or schizophrenia. Thank you.
Yes, so this is a misunderstanding. I'm sorry. So there are lots of papers that link neurexins and neuroligands, which are molecules we discovered and we studied, to neurodegenerative diseases. I'm rather skeptical of these studies, and we've never published anything like it. But there is an enormous number of studies that show that mutations in neurexin 1 in particular, but also in neuroligands, are major factors in neuropsychiatric diseases, so that linkage does exist. And we have worked on this extensively. It's against the subject I didn't have time to discuss today. And there clearly is it's one of the best evidences that schizophrenia and autism actually does involve synapses as a primary point of dysfunction. In Alzheimer's disease, the way I think about synapses, which is exactly what I'm interested in, which is the relation of A-beta into synapses and ApoE, I think that synapses in Alzheimer's disease become dysfunctional because synapses are inherently fragile. And again, that goes back to the basic biology, to some ways to common sense. Synapses are formed by neurons far, far away from the cell body. They are something of, it's like if the cell body was the size of let's say, the campus of your university here, the nerve terminal could be on the moon, okay? Just to give you an idea of the relationships of the sizes. It's enormous, and that needs to be continuously supplied, continuously kept alive. So I think the fact that synapses are lost in Alzheimer's disease reflects the vulnerability of synapses because they're just simply more fragile. And I believe that the key to the disease might be, I don't know, okay, so let's keep this in perspective, might be to understand better how to make synapses more resilient so that they don't degenerate, and as a result the patients could maintain cognitive functions longer. That could be a rational approach, but in order to pursue that approach, we actually need to know why the synapses are damaged. What is the actual toxic insult? What is the toxicity there? And so that is a key question that requires basic research first.
So, you want to comment? Yeah.
Because there are many psychiatric students in there. So why I comment that this psychiatric disease is a neural dysfunction disease. So far as we know, Alzheimer's disease, about one neuron, normal person, one neuron has 2,000 mitochondria. But Alzheimer's disease only 8 or 8 to 400 mitochondria. Those are 1.5 of mitochondria. So there are many psychiatric diseases can be used BDNF treatment or use mitochondrial treatment, such as autism, depression. So there are many, in China Medical University Hospital, we have 3 platforms. One is small molecule for Alzheimer's disease. Another is EV, exosome. Exosome contains BDNF microRNA to treatment of depression, treatment psychiatric disease, even the schizophrenia is also. So another is a platform is mitochondrial transplantation therapy. Now it's just ongoing 3 platforms. If you have, if you want to know our process, you can contact with us. We will pay more detail for you.
Okay, I'd like to take this opportunity. I think it's almost 12:10 now, so the time is almost up. But we actually have almost 40 minutes discussion. And I'd like to take this opportunity to thanks for Dr. Südhof coming to Taiwan. Please come more often. And first one, I'd like to thank all the panelists this morning. I think we learned a lot about neuroscience, and also to all the audience for your interesting, for your stay and for your participate in this activity. And then certainly President Chai and the International Peace Foundation outside.
Before the end of this section, we would like to invite our president. He wants to give a gift for all of the panelists today. So how about
gift? Thank you.
Professor Südhof? We need to take pictures, so come in the front. 可以到前中间拍了。哦，对对 ，OK，just have a photo together. Do you want to join? Come here. Get this side and other 廖主任你come here， 过来一个，廖主任过来这边，不然的话那边太多了。来这边。Okay. Yeah. Okay. And the other panelists just stand right here. Do we need a good photo? I guess we have that. How can we have a good photo for everybody? Please back on your seats. We want to take a good photo. 回到座位，我们来。